how they work. So we're going to talk through these. So these are called Baltimore classifications. Not everybody classifies viruses this way, but I think this is a cool way to do it. And he won the Nobel Prize for his work in figuring out an enzyme that comes with viruses called reverse transcriptase. We're going to talk a lot about reverse transcriptase and what it is and what it does. That's a viral enzyme. So we're going to talk about, remember, viruses sometimes will come with an enzyme, and one of those enzymes is called reverse transcriptase that we'll talk about. I think he works at Berkeley or Stanford? Stanford? I think he works at Stanford. I don't know. Super cool guy. Still alive. And yeah, just groundbreaking. Okay, so these are the classifications, but don't freak out because we're not really going to memorize any of these. But I want you to understand fundamentally what we're talking about, because I want you to understand what is something called the central dogma. I want to talk about the central dogma. I don't really want to talk about that yet. That is a term that I will have you guys understand and know later on. But what I want to talk to you guys about right now is let me just put it up here because I'll walk you through my thoughts. Central dogma. What the central dogma is, Watson and Crick came up with this term. It's DNA to RNA to proteins. So what that means, this is just everything we already know. We know that. It just has a term. The term is central dogma. So it just means this is how all life works. But viruses aren't alive. They get to kind of free will do what they want. So this is central dogma. And what it is we're talking about transcription and translation, right? Going from DNA to RNA to proteins. Well, don't forget there's this other little offshoot, which is called replication. So not only do our cells need protein to build, we need enzymes, we need all of that, but also we need replication. And replication goes from DNA to DNA because we need to fill our new cells with new DNA. So that's kind of two concepts that we're going to be talking through. Okay, so here, so we're going to talk through a couple of these examples of different types of viruses and how they go through these processes and what they do. So this is a weird thing that different viruses do. So here's class 1 viruses. You don't have to memorize any of this. I'm just going to walk you through what you need to know. We're going to just kind of walk through it so that you fundamentally understand it. So class 1 viruses, for example, papilloma viruses like HPV or herpes viruses, adenoviruses, these viruses are double-stranded DNA. And that makes sense to us, right? Because we are double-stranded DNA. Everything on Earth is double-stranded DNA. That makes sense to us. Well, adenoviruses, class 2 viruses, adeno-associated viruses, these are adeno-associated viruses, these have single-stranded DNA. In our meaning of life, in our organisms on the planet, there is no such thing. Only viruses are able to do this, have a single strand of DNA. Double-stranded RNA, super weird, right? There is no such thing as double-stranded RNA in the life as we know it. Bacteria, archaea, humans, plants, animals, there is no double-stranded RNA. Here's positive single-stranded RNA. So what positive single-stranded RNA means is that when you have your double-stranded DNA, Then you separate it apart. So that your RNA polymerase can get in here and make some RNA. Your RNA polymerase is going to come and copy this template strand, move along this way. And here is our RNA strand. So this is a positive single-stranded RNA. Positive single-stranded RNA starts with a 5 and ends with a 3. The reason is because RNA polymerase reads towards the 5, right? Everything reads towards the 5. It's reading towards the 5. So it makes this 5-3 RNA strand. This is the beginning, this is the end. So this is a normal positive single-stranded RNA. Starts at the 5, ends at the 3. This is actually called a cap, and this is called a tail, head and tail. Yeah?
So this is kind of the extension of the RNA transcription that we see from the transcription bubble, like when it's coming out and it's turning into the mRNA?
Yes. Mm-hmm. When that little tail comes off in that RNA, we have that RNA piece. This is the RNA piece.
Oh, okay.
Yes, yes, yes. So this is A's, U's, G's, and C's. Yeah? Okay. So here is a positive single-stranded RNA. Well, here's a negative single-stranded RNA. That means it's backwards. So some viruses have backwards RNA. Some have single-stranded RNA and they come with this little enzyme that's called reverse transcriptase. So we're going to figure out what that does. So I'm going to walk you through a couple of examples, and so that you kind of understand just fundamentally what viruses do that are different. So we're trying to understand how viruses do it differently than we do it. So one thing that's cool about a virus, that's cool for them, bad for us, is that viruses, because they don't come with very many things, they don't come with a way to make ATP, they don't come with a way to do anything. They don't come with their ability to do anything on their own. So they hijack our cells and use our cells for everything they need. So we're going to figure out how some of these viruses get away with using all of our polymerases, ribosomes, to make all of their own proteins. But then we're going to figure out how some viruses do it differently. So let's walk through it. Okay, central dogma, we talked about that. Okay, so let's talk about class 1 viruses. So class 1 viruses are double-stranded DNA. So let me walk you through this. So here's a virus. Here, let's say this is an enveloped polyhedral double-stranded DNA virus. So here's the envelope, here are the capsids, here's the double-stranded DNA. Well, let's talk about a human cell. Here is a respiratory cell in the throat. Well, what this virus is going to do, remember it has spike proteins, this virus is going to attach to this cell. And so these spike proteins are all over. It's going to attach to this cell, and these little spike proteins are what trick the cell into letting it in. They're protein signaling that says like, oh, yum, food. And it tricks our cell into letting it inside. So now what's going to happen is this cell is going to start enveloping this virus. So here's the virus inside, and here's the virus inside. So now the virus made it inside the cell. Well, what happens is our cells, how we eat the food that we've taken inside the cell, is that we bind it to another pocket of enzymes that are going to break it up. So we have these little pockets of enzymes that break up food. So now we're trying to break up this food. So what happens is we break up the virus into tiny little pieces until the only thing that's left over is its genome. So now the genome of this virus, here's the double-stranded DNA of this virus. Here's our cell. Now this breaks open. And what's left over are all these tiny pieces of the virus plus the viral genome, the DNA. Draw it nicer. So here's the viral DNA. Well, this viral DNA, if it gets into our nucleus, we're going to replicate that as if it's human DNA. We can't tell the difference. DNA is DNA. So what our cell is going to do is start replicating this and transcribing it and translating it. So we're making the viral proteins. We're making the viral DNA. We're making the viral RNA. All of our enzymes can't tell the difference. All we do is just replicate what's inside. So now inside of this cell, we have made tons of viral DNA. And we've made tons of capsids that are going to start forming around these viruses. Because we made all of those. So if track this with me. A viral capsid is a protein. So on the viral genome, it has to have the gene for a capsid, right? Are you guys with me on that? So you have the DNA of a virus. The DNA codes for viral proteins. So here we've made all these capsids. We replicated the genome because we have DNA polymerase that can read DNA. We have DNA polymerase that can read DNA and make new DNA. So basically what they've done is they hijacked all of our machinery to make more copies of themselves. Let me see if I can think of an analogy. Like if you have this big report due for class, but you have no materials, so you're going to come into the copy room, use the computer, use all the paper, use the stapler, use all the ChatGPT that you use to write your report, use all of these things, you print it out, staple it, and now you can make 1 million copies of those because you used all of the machinery that was already there. So that's what viruses are doing, is they're using all of our DNA polymerase, they're using our RNA polymerase, and they're using our ribosomes to make more of their own proteins and their own DNA. I don't know how you guys are doing. No idea. Thumbs up? Thumbs up? Middle? Kind of middle?
I got kind of
Middle?
like on the positive ssRNA, when you went into that strand, I kind of got a little...
Yeah, let's talk about that in a minute, but great, thank
But
you.
everything
I will
else-
go back over that. Okay, I'll go back over that when we talk about what those viruses do. Yeah, but let's talk about that. Okay, so let's just focus on, this is our double-stranded DNA. Our double-stranded DNA is going to get inside. We have the enzymes that will read double-stranded DNA, because we have double-stranded DNA. So it's just gonna read it as if it's our own. It gets into the nucleus, and we're gonna copy whatever's there. DNA polymerases are just proteins that read. So now we've made the viral genome, and now we've made some capsids. Our ribosomes read that, our ribosomes assembled those. Now what they're gonna do is, so now once all of these capsids are assembled around the genome, so watch this. Here's our capsids. Here's our capsids. Except we've made thousands. Now it's gonna start fusing. Actually, what we do is start throwing out spike proteins. Spike proteins go and fuse with the end. Now this virus is going to start fusing with the end here. So here comes the helical capsids, here comes the double-stranded DNA that we've replicated. Now what it's going to do is move out, should get like this. Here are the spike proteins of the virus. Here are the capsids. Here's the double-stranded DNA. And now it has your envelope with it. Now it's taken your phospholipid bilayer with it. All of these are going to take all your phospholipid bilayer with them and go out and infect another cell. So it gets in, dumps its DNA. We replicate, transcribe, and translate that DNA. We've formed a virus inside our cell, and that virus will leave and take our phospholipid bilayer with it. So basically, we are a viral making machine because they just use all of our pieces. So that is a class 1 virus. Double-stranded DNA, copy the double-stranded DNA, and we can make the whole virus, and the virus leaves the cell. So let's talk about class 3 viruses. So class 3 viruses have RNA. They're RNA viruses, double-stranded RNA viruses. Well, let me mute this really quick before this confuses you. Okay, so we have, let me see how I want to explain it. Okay, so if the virus gets into the cell, let me use my same colors. If our virus gets into our cell and it has DNA, this is viral DNA. Here is double-stranded viral DNA. Here's our nucleus, and we have double-stranded DNA. So these viruses are stoked because they can already just use our DNA polymerases, use our RNA polymerases, use our ribosomes, and they can replicate themselves. Well, if we have a virus that gets into the cell, here's our own DNA, but this virus is an RNA virus. So here's RNA. And this is the virus. How does it copy its genome to make more RNA? I'm going to give you guys like 2 minutes to talk about that with the person next to you. Talk about that together, talk about that together. You 3 if you got 3. You 2, you 2, you 2. You 2, you 3 back there, you 2, you 2, you 2, and you 2 back there. Talk about how a virus, talk about the problem. What is the problem with this virus? Talk through it a little bit. What is the problem? Yeah.
Sorry, can you repeat the question?
DNA.
Yes, absolutely. So if a virus has RNA, how does a virus replicate? How would an RNA virus replicate its genome if it has RNA? It's kind of a trick question, but just kind of think through it a little bit. I just want you to kind of labor through, like, what is she talking about? Where's the RNA coming from? What does RNA? We replicate RNA. What is that? What do we need? Just talk through that. Talk through it with your partner. Talk through it with her. What does RNA polymerase do? So talk through that a little bit. What does RNA polymerase do?
Like the enzyme's gonna eat through for the RNA to enter, and then
It produces a new viral genome and messenger RNA
Is it in phase?
to protein synthesis or protein synthesis.
Like that, and then it enters the cell,
Can reshape.
and then
It's kind of like that function.
yeah,
oh,
it starts, replication, and then.
I had a feeling it had to do with RNA polymerase.
interesting.
It's enzyme.
But it needs
I think I was just thinking that with bacterial DNA, where it kind of needed the polymerase to start, but here it's to kind of eat away at it, and then Did you guys turn in your notebook? Did you guys turn in your notebook?
Yeah, those so class 3 virus.
Actually, I'm not gonna be that crazy.
And then RNA transcribes.
It was an archaea, not a bacteria.
I didn't really go over our
Yeah, I think that's what we're gonna get to. Oh, sure. I don't know.
the first lecture she went over really briefly in bacteria, but yeah.
Okay, let's talk through it together. Okay, so an RNA virus, so here's a human genome like normal. We have normal DNA that goes through normal replication with the DNA polymerase. Now we have another set to get into our cell number 2. Also, what we can do with that DNA is that we can read it and make some RNA, and we can translate it and make some proteins. This is normal human setup. Well, a virus that has double-stranded DNA is going to use our DNA polymerase to replicate its own genome. It's also going to use our RNA polymerase to make our own RNA, to make its RNA, and it's going to use our ribosomes to make its own proteins. So this is an example of a class 1. Well, class 3 has this. So now it's weird, right? You're like, well, it skipped this whole step. So how can this RNA make viral proteins? What is it going to use to make proteins? Normal ribosomes. Beautiful. Okay, great. So this is human ribosomes. Okay, so now it can make RNA. Now it can make viral proteins. So this is viral RNA. And this is viral proteins, but this is where I'm getting at. This is why I like talking about Baltimore classifications, because it helps you kind of think through our enzymes and what they're doing. So this viral RNA is gonna make some proteins, but now what? What's gonna go inside of the new virions? What's gonna go inside? If you're gonna copy your genome, if you're gonna make new viruses, what do you need more of? I'm kind of asking them, sorry, that's me. What do you need more of? What does the virus need more of? So they need more proteins and they need more RNA. Because without their genome, I'm gonna get some candy out. Dr. Erica is okay. So for a virus to make more viruses, it needs more RNA. How do you make more RNA? Okay, how do you make more RNA? Any wild guesses?
Transcription, translation.
So this is what I wanted you to think through. You wouldn't know the answer to it, but I want you to think through it, and I want you to use your little thinkers, because this is the problem with RNA viruses. And I kind of wanted you to labor through that a little bit and kind of think through, like, wait a minute, well, how does that make sense? And think through our enzymes, because if you had a new virus, so here this virus is a yellow virus, it has the proteins because it can make proteins. It has proteins because it can make proteins, but how does it get more RNA? Here's another one. We have RNA to make proteins, and we have RNA to make proteins, but how do we get more RNA to make new viruses from not having any DNA? There is no DNA available to make that.
Well, doesn't it use the human cell to
So this is a fantastic discussion. Keep the questions going, because I have to work you through it, but I think all of these questions are a good point. 2 seconds, really quickly, because Dr. Rowe had a good point, sorry, what was your point again?
I said,
DNA.
well, if it invades, then it uses human cells and we have DNA.
So good, such a great point. We have DNA, but we can't make viral RNA or viral proteins because we don't have the recipe for that. Does that make sense?
Yes.
Okay, cool. Yep, let's keep thinking through it. Yeah.
Could it like encode for a new enzyme that would make RNA?
Fantastic point, and yes, fantastic point. Yes, so there's a new enzyme that we're going to introduce, but I like thinking through it. I like laboring through these hard questions because it helps you to remember what the virus is doing and how the virus reacts and comparing it to our own genome. So what some viruses have is a special enzyme that makes viral RNA, more viral RNA. And this special enzyme is called replicase.
At least that one's self-explanatory.
Yes, replicase. Replicase replicates RNA by reading RNA. So walk through this with me. We've talked about 3 enzymes. We talked about in humans, I'm going to keep my color coding the same. In humans, we have DNA polymerase. What DNA polymerase is, it's a DNA dependent. Am I spelling that right? Seems weird now. Ant or ent? E. Dependent.
But it's 8.
Why did it look weird?
Like, cause is it like independent?
Okay.
We don't judge for
know.
silly.
Okay, somebody Google it for me. Okay, I don't know why it looks weird all of a sudden. Okay, so DNA dependent, it's a DNA dependent DNA polymerase. It polymerizes new DNA by reading DNA. RNA polymerase. Talk to your neighbor about it 1 second and see how we would describe an RNA polymerase. Talk through it with your neighbor. Even if you're wrong, it's okay. Talk through it with the person you just talked to. What would we put there?
DNA polymerase and RNA, or DNA to RNA and RNA
Do you want to go for it? Go for it.
It's just, it seems like
It's what I said earlier.
Yes, it is what you said earlier. Yes, yes.
like it makes a new enzyme.
Okay, so what do I put right here? What dependent? This is the tricky part. What dependent?
It's also DNA dependent.
Yes, this is DNA dependent RNA polymerase. It reads DNA and makes RNA. Yeah.
So it's DNA dependent because it reads DNA?
Yes. It has to read DNA. It can't read anything else. Does that make sense? It reads the DNA to make RNA.
And then replicate.
So, yes, replicase. Okay, talk to your neighbor really quick.
Oh, we skipped. We already.
What is replicase? Talk to your neighbor 2 seconds. What is replicase?
Well, that's what we said. Yeah, because I thought once you wrote that.
So we're gonna use the RNA polymerase initially. Or the RNA polymerase. No, in the beginning, RNA polymerase is initially DNA dependent to create the RNA, and then from that, replicase will replicate, so we can make more.
It needs.
Okay, anybody want to give it a go? What is replicase?
Oh, okay.
It uses the ribosomes to make the replicase, and then the replicase goes back and reads the RNA to make more RNA.
Yes, so RNA dependent, but what would we say? RNA dependent RNA polymerase. It reads RNA to make RNA. You see some furrowed brows. RNA dependent RNA polymerase. It reads RNA and makes more RNA. Does that make sense? Yeah.
So, RNA dependent RNA, so it's depending, so the RNA is depending on the polyp?
This one dependent means RNA dependent. It means it must rely on RNA to make more RNA. It has to read the RNA to form more RNA.
So, replicase is depending on RNA polymerase, and then,
No, replicase is only so replicase has to read RNA to form more RNA. It doesn't have anything to do with RNA polymerase. RNA polymerase is a whole different enzyme.
Oh.
So if we were to put, for example, how I gave you guys the original example, the RNA, then a lot of people, I heard you say, which I probably would have guessed that as well, is RNA polymerase. Because RNA polymerase makes RNA, but RNA polymerase can't read RNA. It has to read DNA. Right? Only a viral enzyme called replicase can read RNA and make more RNA from reading RNA. Do you guys want to talk through that another second? And then we're going to move on to a new funky one. Okay. See if that makes sense to you.
But I just think she said that RNA polymerase doesn't like isn't involved in the
Oh, I think she just meant like when you're thinking of it like they
Professor?
were together, but they're not the same. So if you think back
Professor?
to the like that, like if you think back to our remember when she had it? Oh, I think this one she did the extra business. She did this. Oh, okay. She wrote like remember DNA to DNA is replication, and then that's DNA polymerase, and then RNA to DNA or DNA to RNA. DNA to RNA is RNA polymerase.
Okay, we have another option of another type of virus. Okay, this is our last virus type, and I'll walk you through it. Okay, so now we have RNA. This is viral RNA. It can use our ribosomes to make proteins. Some viruses use this enzyme called replicase to make more RNA so that it can replicate. Some viruses have this scary enzyme that's called reverse transcriptase. And what it can do is make more viral DNA. So talk through that when we add it here. Talk through that with your partner. Which one is that? Here, if I put reverse transcriptase here, talk through that with your partner. If you get it, explain it to your partner. If you don't get it, we'll kind of labor through it together.
This one needs viral RNA.
Is this also what?
And makes DNA.
No, this is a different class. I'll show you in a minute. Yeah, this is a different class.
You won't get viral
This is
DNA.
class 5. Class 5.
Oh yeah.
Class 6.
It's like
This is class 6.
in a factory, you're making like these beautiful like person dolls or something.
But you don't have to remember the
And then
classes.
you go in there and you have the factory and instead of making this little doll, you're like
This is found.
yes.
Okay, what would we put here for reverse transcriptase is a what?
Yes.
RNA dependent what? Nice. Yeah, you're getting it, you're getting it. If you still are totally lost, I don't blame you at all. Just keep laboring through it, you'll get it. So RNA dependent DNA polymerase. This is reverse transcriptase. Reverse transcriptase we worry about. DNA viruses in general we worry about. RNA we do as well. We'll talk about why some of those differences are. But here, look what this virus can do now. Now that it has DNA, now it can use our DNA polymerase to make more viral DNA. Now it can use our RNA polymerase to make more viral RNA. Now it can use our ribosomes to make more viral proteins. So basically this one has like unlimited options. So now that it has the viral DNA in there, now it can hijack all of our machinery. So these ones are scary viruses. Okay, so I'll walk you through, oh, I can't walk you through the slides quite yet, but does anybody want to take a picture of that, if they're picture people? I don't know if you're a picture person, if that makes more sense to you, but I have them on the slides, kind of. I didn't do it, I think this makes more sense sometimes drawing it out like that. But if you are lost, listen to this lecture again. Record, listen again. Okay, so I'm gonna erase all this and we are going to, I'll show you on my slides what we're gonna do, what we're, how you can study for it at home and what you need to know at home. Okay, so definitely, so what I want you to know, one thing I want you to know are the viral enzymes. I want you to know what replicase is and what it does, and what reverse transcriptase is and what it does. Replicase reads RNA and makes more RNA. Reverse transcriptase reads RNA and makes DNA. Okay. So here, we should pull it up again on here. Okay, so you don't have to remember the classes, I just want you to understand how viruses work. Viruses can use our human polymerases to make more DNA. But we can't use DNA polymerase to read RNA. We can't use RNA polymerase to read RNA and make more RNA. We have to use this called viral replicase. So we use replicase to make more RNA. We use more replicase to make double-stranded RNA. Now it can fill its little capsids with the new double-stranded RNA it wants. So I wanted to say really quickly also what class 5 viruses can do, but just stay with me for 2 seconds, but you can leave this slide out if you want to. But when we have one strand, so RNA, we haven't talked about this yet, really. Okay, RNA goes this way. This is called the cap, and this is called the tail. Just stay with me for 2 seconds, and then I'll just tell you what you need to know. RNA goes this way. This is a positive RNA. Well, if a virus comes with a negative RNA, it has to be turned into a positive RNA because this is what our ribosomes will read. Our ribosomes are going to come on here, and remember, here is our start codon, AUG. It has to start here. So if you have a backwards RNA, our ribosome is going to attach here, and that's it, and fall off. That's a backwards RNA. That's a negative RNA. So replicase also will read this and make a positive RNA from it so that our ribosomes will read this and make proteins. So what I want you to know from that is I want you to know that positive single-stranded RNA is what we have. That's what we use. That's all I want you to know. You don't really have to know the difference between positive and negative, and if you don't want to, if it makes sense to you and you're like, cool, it makes sense, more power to you, good. If you don't want to and you're like, okay, I'm already kind of hanging in there by my fingernails, then just ignore it. But all you need to know is positive is what we have. We are positive. Positive is our RNA. It goes in the correct direction. Positive means right. Okay. And by right, I mean correct, not directional.
Humans are viruses.
Humans are positive. Okay. So some viruses come with a backwards RNA, and replicase can make it a forwards RNA so that our ribosomes can use it. Side note. Okay, so reverse transcriptase to DNA. Now we can use human RNA polymerase to make RNA. Now that we've gone from RNA to DNA. So walk through these at home. If you need me to walk through them with you again, I'm happy to. Okay, so here DNA-dependent DNA polymerase, DNA-dependent RNA polymerase, RNA-dependent DNA polymerase, RNA-dependent RNA polymerase. Those are the viral enzymes that we need to know. Okay, oh, do you want to take a picture of that? Here, yeah, you can always tell me, I'll pause it. Okay. So viral transcription and translation happen with human ribosomes. Viruses don't have ribosomes. Yeah? Okay, so let's talk about mutations. So there's only one main point that we need to know about viral mutations. And this is the main point. You don't have to memorize any of this. Let me just tell you one overall arching idea of viruses. So why, when we get a vaccine, do you guys know why we have to get a flu vaccine every year, or you have to as a healthcare practitioner? Why would we have to get one vaccine for HPV and one every year for influenza?
So you don't miss
So it's because they're always changing. The virus constantly changes, always. So scientists are always trying to come up with new ideas of what we think, the ways that we think the viral genome will mutate. Because mutations are really, really common in viruses. But DNA viruses generally, this is not a hard and fast rule. It's just, I want you to remember that DNA viruses mutate less often. RNA viruses mutate more often. But I also want you to remember it's not a hard and fast rule, that it depends on the virus. But generally, that's what we think. So, can somebody guess, this is definitely a candy question. Maybe you need to talk about it with your partner, because it's a tricky one. Why do you think, that was for your last one, good job. Why do you think that viruses that are DNA viruses are less likely to have mutations than RNA viruses? Think through it with your partner.
I think it's
because the
DNA we have
You had to
stop it versus RNA kind of takes over with things like reverse transcriptase. That's my guess. Is that the RNA transcriptase kind of goes wrong?
This is a tricky one. This is really tricky.
I don't know.
I think the polymerase doesn't check for any errors.
Because it's double stranded.
And then, if I remember, it's like
That's a good
2 or 3 different like people, little whatever they call them, people. Because that's what I memorized. It's like before they start all the things.
You're saying the right thing.
Checks for errors and then
You said it too.
yes.
You earned it. You earned it. You earned it.
That proofread, yes.
Oh, I heard the word. I heard the word. Did you guys say it together?
Yes, I accidentally grabbed 2
Oh,
earlier.
yes. Oh, good. Okay, so we had it up here. Does anybody know why? I heard the magic word up here. Yeah, go ahead.
They like replicate differently.
They do absolutely replicate differently. Yes. What is the key? Yeah, go ahead. So that is true also. The genome is smaller. It's more like temporary. Yes. What else? There's a magic word I've been thinking of. This front group had the magic word. Keep thinking through it for a second. Anybody else have the magic word? Okay, you guys go ahead. Proofreading. So DNA, when the human genome is replicated, we have this beautiful enzyme that repairs our genome. We have repair polymerases. Like in bacteria that have DNA polymerase 1, we also have repair polymerases. So when we go from DNA to DNA, we have an enzyme that goes through and double checks everything, that proofreads. We don't have anything that proofreads RNA. We don't really care if RNA is broken. If RNA is broken, we have one bad protein. If DNA is broken, we have every single protein bad from there, the entire life of that cell. So proofreading, that's the main reason why. So RNA viruses are more prone to mutations. DNA viruses are less prone to mutations as a blanket rule, but not always. Okay, zoonotic diseases. I'm gonna play this for you at one point. I'm not gonna do it yet, but I'll play for you a little segment of this book because she talks about zoonotic diseases and how prevalent they are and how prevalent they're going to become. So this is a beautiful book if you ever get a chance to read it over the summer or something. And this book I really like a lot actually because it's so inspirational. You know, our world right now, I'm part of a group, it's like a, I don't want to say like a meditation group, but something like that. Like we talk about kind of like the deep ideas and what we go through and all that kind of stuff. But there's a guy on our group that is constantly talking about like how terrible the world is. And he's like, Epstein and Iran and like all of this horrible things that's going on in our world. And this book is so inspirational. So if you need some like food for like, okay, let's get moving. Okay, we can do this. Okay, the world is not going to fall apart. So this is the book for that. It's so good if you guys get a chance to listen. If you listen to it on Audible, I like it on Audible actually better because it's her voice and it's Douglas Abrams. Douglas Abrams is a New York Times reporter. And they talk back and forth, and so it's kind of like podcasty. It's really beautiful, and she's so amazing too. So, if you get a chance to listen to that over the summer, but I'll play a little portion of it for you. So she talks about in that book about zoonotic diseases. So zoonotic diseases are really common from viruses, but we also get zoonotic diseases from all the other microorganisms that we talk about all semester. But we talk about it a lot when we talk about viruses because viral zoonotic diseases are terrifying, as we know, because this is what caused the COVID-19 pandemic, is a zoonotic disease. So let me tell you why COVID-19 happened and why it got so bad. So the reason it got so bad is because something called antigenic shift. So antigenic shift is when in an animal, birds get viral infections all the time. They get respiratory infections, they get viral infections. They pass it between chickens, they pass it between different types of birds, and they'll pass it around. Well, we get viral infections, different types of viral infections, and we pass around our viruses, and we pass it around. Well, when a zoonotic disease happens and these two come together, this virus that the bird had and this virus that the human had, when those jump over, now our body hasn't seen anything like it. It's an amino acid combination our immune system has never seen. So our immune system hasn't built up any resistance to that. So it's something totally wild and foreign. So that's what happened with COVID-19 was they think bats and pangolins had this virus. Well, when people eat bats or eat pangolins or in close contact with them, or close contact with birds, close contact with pigs, like swine flu, bird flu, things like that, then they can catch these viruses that are so foreign to our body that our body has zero immunity to it. So the virus basically is like running the show, and our immune system is like, I don't know what this is. It takes us forever for our immune system to try and catch up with it. And it's highly virulent. It has a lot of bad cytopathic effects. So that is what caused COVID-19, was antigenic shift. Antigenic drift is what happens within a normal population. It's drifting. It's like lazily drifting. Little tiny mutation by little tiny mutation. But then it's drifting over there as well. Little mutation by little mutation. But then you get the two together, and now it's shift. Now it's something so different that our bodies haven't seen it before. So it's a big change because our bodies don't recognize it. So antigenic shift are little mutations over time. Antigenic drift is like somebody who's drifting, who's lazily moving along. That's little tiny movements. Shift is a big giant movement. And one big giant movement usually happens when zoonotic diseases, when viruses jump from species to species. So that is, so you can watch this little video. This talks about H1N1, it talks about the different viruses. We'll talk more about that as we go along. But these are zoonotic diseases. We worry about antigenic shift a lot. Yeah.
How are they able to make vaccines so quickly for shifts like that?
Great question. So that was a big, I know you remember it, I'm sure, in the news that it was wild, the whole mRNA vaccine situation. So we'll talk about that when we talk about vaccines, but fantastic question is, we made a really fast vaccine because we had to allow for mRNA vaccines for that to occur. So yeah, really great point. So we're gonna talk about mRNA vaccines, yeah. Okay, so these are a lot of the different zoonotic diseases. I made this slide when monkeypox was going around. I don't know if you guys remember that from a few years ago. There was a big old scare about monkeypox. So yes, really gross. So that was a zoonotic disease. Now we don't hear about it as much anymore. Rabies is a zoonotic disease. All different types of influenzas. So yeah, we'll talk more about zoonotic diseases as we go along. Okay, I was gonna talk about life cycle viruses, but I think I'm gonna skip it because I want to cover cytopathic effects. Okay, so I am gonna talk about, you know what, I'm gonna do it because there's only like one thing I want you to memorize. It's just tiny, so let's just do it really quick. So the tiny thing, so we're not gonna memorize any of this. Life cycle of animal viruses. A lot of micro classes will want you to memorize it. I think it's just kind of self-explanatory, so I'm not gonna have you guys memorize it, but let me just tell you what I mean by self-explanatory. The virus attaches to the host cell, it gets into the host cell, it replicates, and then it leaves the host cell. These are just all fancy terms to say that. So instead of memorizing any fancy terms, we just know a virus attaches to a cell, it gets into a cell, it replicates within the cell, and then it leaves the cell. So we don't have to memorize anything. Just makes sense, right? This is what viruses do. They get in, they get out. But a couple of things I just wanted to mention really quickly is remember that there's something called tropism. If we breathe in HIV, we won't get it. It's not respiratory. If we sit on a toilet with COVID, we won't get it. If we even exchange blood with a patient that has COVID, we won't get it. It's respiratory. So different types of organisms have organisms, or don't let me call them organisms. Different types of viruses have different tissue types, different tropic tissue types that they prefer. That's the cell type that they're allowed to attach to that they can get in and replicate. So that's just another term that I wanted to bring up. And then, so they get in, they get out. That's it. So these terms that I'm gonna bring up really quickly, but I'm not gonna have you guys memorize these terms either. Why do I not want you to memorize these? I don't want you to memorize these because this is what you're gonna see every time you Google life cycle of a virus, lytic and lysogenic, but that's actually only for bacterial viruses, only for bacteriophages. Human viruses, we call it the productive phase and the latent phase. So we'll talk more about that when we talk about cytopathic effects. But just so you know, so what a lytic cycle is when the virus is replicating. It's called the lytic cell, and it lyses out of the cell. The lysogenic phase is when the virus is hiding inside the cell and stays dormant. So just forget all of this. I won't ask you any questions about life cycle of a virus, so we'll just skip it. Just so you know, it gets in and gets out. Oh, I am going to show you this video really quick though, because it's only 2 minutes, and it does a really good job of showing it to you.
You probably already know that viruses are responsible for causing a number of diseases, from AIDS to the flu. But did you ever wonder how a virus works? Viruses are basically infectious particles that take over the operation of a cell for the sole purpose of...
So watch how it just did that. It did it better than I drew it up on the board. Let me show you really quick.
Viruses are basically infectious particles that...
So see how the virus, the spike proteins attach to the cell and trick the cell into letting it inside.
that take over the operation of a cell for the sole...
Now it empties its genome into the cell, whether it's DNA or RNA. If it's DNA, it gets into the nucleus. And now we're replicating its genome, we're making its own RNA, and we're making its own proteins. So now all the virions, in our own cell, we've made all the viruses. We've made all the virions.
manufacturing new viruses. How exactly does a virus do this? In many ways, it works in the same manner as a computer virus does. It has to first gain access and then convince the machinery within the device, in this case a cell, to make multiple new copies of the virus. There are many different types of viruses, but they do share some similar characteristics. First of all, viruses are usually specific in the types of cells that they infect. The specificity of a virus is dependent upon the types of receptors that are found on the surface of the target cell. Every cell in your body has a pattern of protein receptors on its surface. The virus uses these proteins to target specific cells for infection. In this example, the genetic material of the virus infecting the cell is DNA. However, unlike the complex DNA found in the nucleus of a cell, the DNA of a virus is relatively simple and just contains the information needed to manufacture new virus parts. Once inside the cell, the instructions in the DNA are transcribed to RNA. The protein building machinery of the host cell then translates these instructions into the components of a new virus. These parts are then assembled into new viruses within the host cell. When ready, they emerge from the host cell, often killing it in the process. As they emerge, some viruses retain parts of the host cell membrane, forming an envelope around the virus. This envelope gives some protection to the virus from the immune system of the host organism. Each new virus is now capable of infecting another host cell and repeating the process of virus replication. By understanding how the life cycle of a virus works, scientists have been able to develop antiviral drugs that target specific points in the virus life cycle and thus prevent the virus from replicating.
Okay, so that kind of described what we already talked about, but I thought it was a cool visual. Okay, so let's talk about cytopathic effects of viruses quickly, and then we're going to move on. So how viruses harm human cells. They harm human cells. One, there can just be so many of them that they break the cell open, that they burst the cell open, just like this picture that I showed you. So this is 43 that we're at. Oh, no, it wasn't that one. Let me see. Here, let me show you this one. Oh yeah, this one. So that's one cytopathic effect of a virus, is that it can just burst out of the cell because there's so many inside of it. So overgrow and lyse the cell is a cytopathic effect of viruses. Another method is taking a portion of the cytoplasmic membrane with it. So in enveloped viruses, sometimes, but not all the time, that will kill the cell as well. Is when we take that phospholipid bilayer, sometimes that will kill the cell. So taking a portion of the membrane with it. So this one I have a video on. This is called syncytia. And I'll show you a little tiny video on that. And then also what some viruses are able to do is cause cancer to that cell. These are called oncoviruses. So this is a new term for us, oncoviruses. Oncoviruses cause cancer because the virus alters the human DNA. When it alters the DNA, that can harm the cell so that the cell becomes cancerous. So that is called an oncovirus. But also there's types of viruses that will hide, that will just stay hidden within the cell. So some will kill it, some will hide within the cell. Some will hide in these little pockets called inclusion bodies. And some will hide in the genome. That is called a provirus. So there's two new terms for us. Well, three. Syncytia, oncovirus, provirus. I guess four. Inclusion body. Okay, so let's go through those. So 1 and 2, it can either overgrow and burst the cell open, or it can take a piece of the membrane with it and break the cell open. So that's cytopathic effects number 1 and 2. Number 3 is something called syncytia. So I'm just going to turn the volume down and just let you watch that for a second. So this is what some viruses do. Some viruses, because they're coming in and out of cells and cells are connected together, they cause a membrane mess. So they cause all of the membranes between cells to become where it's like one big cell, because they've gone in and out so many times and they've gone in and out where the cells are connected. So now it forms one big cellular mess. That's called a syncytia, like respiratory syncytial virus, RSV. Very common syncytia in the respiratory system and in the lungs. So you don't want a big cell mess in the lungs, right? Because then you're coughing, and it can cause lung damage, and it can cause lower O2 saturation, things like that. So a syncytia is just a big mess of cells. Sometimes we call it multinucleated cells. It's just a big mess of cells. That's all it is. Okay. So these are oncoviruses, such as human papillomavirus, that can cause cervical cancer. So every female in this room, and if you have patients, advise them to get a pap smear and to test regular pap smears to test for abnormal cervical cells. And because if you catch cervical cancer early, a lot of people have HPV, tons and tons of people. If you don't get checked regularly, then it can cause cancer, and then it can cause cancer of the cervix, which is a very harmful type of cancer. We worry about cervical cancer a lot with our patients. Epstein-Barr virus, which is a type of a herpes virus. We'll talk more about that later, about it can cause lymphoma, stomach cancers, nasopharyngeal cancers. Hepatitis B, hepatitis C can cause liver cancer. So different types of viruses can cause cancer. The only thing you need to know is the term oncovirus. But I just put this up here because I thought it was cool. These viruses harm the cells in different ways. And if you ever take a cancer class or learn anything about cancer, it's super fascinating, because cancer is not just one disease, it's multiple diseases of the cell. So different viruses can affect different aspects of cancer, like pre-cancer. So they can cause, for example, something that's called angiogenesis, where a cancer cell that starts a tumor, it needs blood supply. And usually it needs more blood than normal. So it causes the blood vessels to come into the tumor. So sometimes viruses can do that, can cause more blood to enter into a cell. And so that could be a first precursor for cancer. But all kinds of different things that they can do. You don't have to memorize any of that, I just think it's cool. So what an inclusion body is some viruses that can hide. They can hide inside of these little pockets inside of a cell. So they hide inside of little pockets inside of a cell. These little pockets are called inclusion bodies. And then this, for example, these are called proviruses, where the viral genome is actually going to attach or infect our own genome, which is very, very dangerous. That means it's not ever going to go away. So, for example, herpes viruses or human immunodeficiency virus. Somebody with HIV will always have HIV, even if they're on HIV drugs. Because we're trying to, what we can do now is we can prevent virions from forming, but that person will always have that viral genome in their genome. Same as herpes simplex 1, herpes simplex 2, that we'll talk more about later. But those viral genes will attach to our genes, and the person has that forever. So this is just a cool video about bacteriophages, about viruses that infect bacteria. But you can just watch that on your own. So we'll talk more about herpes viruses and HIV later. So for example, so these are what are called latent viruses, and they will remain dormant in the genome. Like for example, I get cold sores, and so it's that the viral genome is on my DNA forever. And then when my immune system is down, or I drink too much booze, or I'm in the sun too much, it starts producing virions again, and then I get a cold sore. So that's what herpes simplex 1 is cold sores. That happens frequently. Herpes simplex 2 is genital herpes, and that's common as well, and that stays forever. So different types of viruses stay in the body forever, but different types don't. If you've already had COVID, SARS-CoV-2, then once it's healed, it's all the way gone. So different viruses work differently. So we'll talk more about that next time. Okay, so what I'm gonna have you guys do is take, just try and take kind of a quicker one if you can, about 10 minutes, and then we will